So Professor Karunakaran is here to deliver his talk on immunobiosensor. I invite Dr. Karunakaran. Please.
Thank you, Dr. Professor Balakrishnan, sir. Let me start the session. Very good afternoon, all the participants and all of you. At the end of this lecture, I hope you will be able to design your own biosensor or immunosensor in your lab. We'll know that few terms, what are called sensors, what are called transducers, because so that you'll be familiar with this topic. A sensor is nothing but an object or device to detect changes in environment, changes in analyte, or changes in chemical concentrations of chemicals that provided output. For example, change in pressure, change in humidity, change in concentration of analyte, change in protein, etc. It is called a transducer. It is a device that converts one form of energy into another form of energy. For example, chemical signal, biochemical signal is converted into electrical signal, like etc. Commonly used to what is called a biosensor. The biosensor is an analytical device which converts a biochemical signal, biochemical reaction into an electrical or optical signal. Commonly used biosensors are glucometer. Everybody will be using. Most of them are using pregnancy test, etc. The principle of biosensor. In biosensor, the analyte binds to the bioregulation element. Action or the biological event is converted into electrical signal. This is amplified further and the signal is processed. What are the applications of biosensor? Two types of biosensor: bioregulation element, bioreceptor, and transducers. Biologists use this one, bioreceptor based. For example, antigen antibody is a biosensor. Enzymatic biosensor. Even cells could be used as biosensor, as we see later. DNA, single strand DNA could be used phage biosensor. Chemist and physicist, they will use these types of, for example, electrochemical biosensors, ambrometric biosensors, measuring current, potential, measuring conductance, piezoelectric changes in mass due to the interaction between antigen and antibody. We can measure the changes in mass, etc. As you see, shown here the cell surface, there are several receptors. Similarly, you have COVID-19. Glycoprotein overexpresses glycoprotein and it binds to ACE2 receptor, causing this disease. This is the biosensing principle I have shown briefly. The most important event is molecular recognition. Is very important. In this case, antibody-antigen interactions and other biological recognition events are more specific. Biosensor is a multidisciplinary field. You need to know chemistry, biology, physics, and instrumentation to develop a device. You need instrumentation knowledge, electronics. Once you develop a product, it should be marketed. Then marketing also you need. Of biosensors, it has wide applications in drug discovery, disease detection, environmental monitoring, toxins in defense area, etc. We are focusing on mainly electrochemical biosensors because of operation simplicity, low expense of fabrication, suitability of real-time detection, fast response, most sensitive, and selective determination. The mechanism of glucose biosensor. For example, there are several beginners here. How glucose biosensor works? glucometer works. I will show you here. Glucose on reaction with the oxygen in the presence glucose oxidation. The enzyme is immobilized on the electrode surface. Okay, in the presence of oxygen, glucose oxidase converts glucose into gluconic acid and hydrogen peroxide. As you see, there are three ways glucose biosensors can be made. There are three ways. One is reduction in the concentration of oxygen. That is Clark's method. This is the first biosensor. And next, you can either follow hydrogen peroxide, the formation of hydrogen peroxide concentration. More the hydrogen peroxide concentration, more the glucose concentration, or measuring the change in pH. For example, gluconic acid is formed. This gluconic acid is acidic. Therefore, increase in pH due to this reaction can be followed. There are several enzymes for each analyte. For each analyte, there are oxidase, glucose oxidase, lactose oxidase, starch oxidase, glutamate oxidase, for example, neurotransmitters. So you can use the same one. If you are able to develop one biosensor, then you can 1 crore biosensor. Very simple. So let us see genosensor, how it works. It is very simple. You take DNA as the biorecognition element. It is immobilized on the electrode, and as due to high nucleic acid hybridization, the change in mass or change the binding event is detected. It is the signal is converted into electrical signals and measured. So the same principle is used in DNA microarray biosensor in several electrodes. Okay. Biosensors, multiplex biosensors are used for detecting mycotoxins in plants, grains, etc. There are several mycotoxins I have listed here. As you see, it can be. This is the multiplex. There are 4 electrodes, working electrodes. So 4 mycotoxins, 4 analytes can be separately determined at one shot, simultaneously determined. So there are several biomarkers. For example, diabetes glucose is a small molecule biomarker, and hypertension, for example, plasma renin, and asthma, and cancer, prostate specific antigen, EGFR, etc. For each disease, for each pathology, there are several biomarkers. Once you identify the biomarker, you can easily develop the biosensor. We have carried out a collaborative research project with the DRDO, where in the high altitude, for example, Siachen and Mount Everest, the oxygen level is very, very low. Therefore, the Jawans, the military soldiers, once they are deputed to hill region, they will hypoxia, that is decrease in oxygen disorder. These are the various high altitude. Suddenly, all of a sudden, a few of them are very susceptible and sudden die. So there are several researchers going in DRDO lab for this. We are also part of this research. So, conditions due to hypoxia at high altitude, reduction in oxygen concentration. As you go higher and higher, the oxygen level, oxygen concentration decreases. Therefore, you will develop hypoxia symptoms. Some of them will die immediately. The hypoxia biomarkers I have listed here. Some of them are small molecules, some of them are proteins, and there are several grease method, fluorometry method, immunobiosensor method, HPLC method. These are the various chemical methods, and there are several biochemical methods for the detection of biomarkers: ELISA, Western blot, flow cytometry. The drawbacks, the major drawbacks are tedious detection procedure, time consuming, less sensitivity, and okay. Now the success of biosensor fabrication is biofunctionalization. The electrode surface is modified and biofunctionalized with the biorecognition element. So biofunctionalization or bioconjugation is an important step in the development of biosensor. I have given some of the for example, nanoparticles are often used for biosensor fabrication because of large surface area and increase in sensitivity and selectivity. The carbon, I have shown here the carbon nanoparticles, how it is oxidized and conjugated to enzyme by chemical method. Okay, chemical methods used for bioconjugation. I have shown here, for example, glutaraldehyde method, acyl chloride method. Okay. Method, chemical method, how it is used to, and glutaraldehyde, how glutaraldehyde we have used mostly glutaraldehyde, how glutaraldehyde is used to conjugate to more to the electrode. That is very important step. Otherwise, the enzyme will be washed out of the from the electrode. Let us see here. You will understand now a case. For example, we have fabricated a very simple biosensor for superoxide ion radical biosensor. I have shown here the fabrication of it. We have taken a platinum electrode, very simple platinum because of high conductivity, and then we cannot directly coat or bind the enzyme. Therefore, we have electropolymerized conducting polymer, conducting polymer like PPY. There are several conducting polymers. We have used polypyrrole as conducting monomer. We have electropolymerized, and then we have incorporated gold nanoparticles, and then with the cysteine, in the presence of cysteine, a is placed on the electrode. Cysteine forms a self-assembled monolayer, and this engaged group couples with the glutaraldehyde, and this glutaraldehyde in turn couples with the engaged group of the enzyme. So we have used the coupling agent glutaraldehyde here, bioconjugating agent. Why we have used gold nanoparticles? I have given here. So this is the mechanism of detection, mechanism of biosensing mechanism. Okay, so here the enzyme is coated, and the superoxide reacts with the enzyme to form oxygen, and then in turn superoxide will react with here to form hydrogen peroxide. This hydrogen peroxide form is actually detected. We have used cyclic voltammetry. It is similar to optical spectroscopy. In optical spectroscopy, you measure the absorbance, light absorbance, or OD. We observe the current is. So we have characterized whether the electrode is immobilized with the enzyme. This peak is due to copper redox, Cu2 plus to Cu plus. In the presence of, for example, in the presence of superoxide ion radical, as see the current response, the peak current response increases. This is due to increase in the current response due to the gold nanoparticle. Without nanoparticle, with the nanoparticle, we have carried out, and we have also generated superoxide radical by enzymatic method and detected the increase in xanthine. In the presence of xanthine oxidase, the superoxide radical concentration increases, the peak current increases, as you see here. Next, we have simultaneously detected both, because superoxide radical and NO is converted into nitric oxide converted into NO2 minus in our system. So we have simultaneously measured superoxide radical and nitrite by using the same biosensing electrode. For example, by same biosensing electrode we have prepared, and we have simultaneously detected both superoxide and nitrite. The active site, this is the copper superoxide enzyme. This is the active site of the enzyme I have shown here. As you see, the active site is very narrow. Enter. Okay. Based on our work in Journal of Chemistry. There are two peaks. As you see here, there are two and radicals. So we have simultaneously determined both. We have in cancer cells. We have detected superoxide radical nitrite ions in cancer cells. Next, we developed high enzymatic biosensor. We have, in the previous study, we have only one enzyme on the surface of the electrode. Here, as you see, we have immobilized, used, in this case, CNT carbon nanotube is used. We have two enzymes here, and measured nitrite. So this is a bi-enzymatic biosensor. We have also developed enzyme mimetic-based biosensor. For example, manganese porphyrin, copper porphyrin. They have active. They act as biomimetics. They have used these biomimetics for the detection of nitrite. As I said, at high altitude, NO level is lowered. In DRDO, we have found that NO level is very much reduced. So, the DIPOS director asked me whether you have a biosensor for nitric oxide, whereas in asthma, it is nitric oxide level is higher. The nitric oxide level, by monitoring the nitric level, we can monitor the hypoxia susceptible soldiers. And the same SOD1 enzyme is used for the measurement of NO biosensor. So, using as you see, carbon nanoparticles and the electrochemical response of time. So, and with increase in NO concentration, we have plotted a calibration curve. We have also studied interferences, because in chemical world, in chemical system, there are no interferences. Whereas in biological system, there are several analytes. So we have used an Nafion membrane. We have coated Nafion membrane so that there are no interferences. Also, they have used nitrate. For example, beetroot juice, millimolar of nitrate. Nitrate actually reduces the hypoxia disorder. Nitrate-rich food or vegetable hypoxia. They have tested beetroot juice, especially the spinach. They consist of very rich nitrate content. Take nitrate. In nitric oxide will be produced. Therefore, you will not have hypoxia syndrome. So, how nitrate our actually there are no specific enzymes in our body, but there are some enzymes, nitrate detectase enzymes in our cavity, oral cavity, where nitrate is reduced to enzyme. How much nitrate is present in blood sample, or in saliva sample, or in urine sample, or in beetroot juice. So we have developed a nitrate biosensor to measure the level of nitrate concentration in beetroot and etc. This is again, we have used the same fabrication method as you know how to fabricate this. The others are biological only. So this is a gold nanoparticle, and this is the bare working electrode. As you see, conducting polymer, it makes the nano composite matrix as nanoporous, so that enzymes can be readily bound and it will not be washed away. This is again linear calibration plot for nitrate concentration, nitrate concentration by grease method, chemical method, and also biosensor method. Both agrees each other. Developed biosensor for cytochrome C. Heme-containing protein, metal protein, it is located in brain space, mitochondria, and shown here. What cancer chain, you know, in the during apoptosis, during cytochrome C is released into the cytosol. Cytochrome, this is this cytochrome C, electron, subtle electron, complex 3 and the complex. Okay, so specifically binds cytochrome C. Similarly, copper 4 also cytochrome C oxidase, cytochrome C reductase binds cytochrome C. Using this concept, we have proposed to DPT to develop biosensor for cytochrome C. We have granted 35 lakhs to develop biosensor, and we have developed the biosensor, which you will see now. Okay, so the cytochrome C is during apoptosis. There are two types of cell death, and cytochrome C is a biomarker of apoptosis. So, cytochrome C reductase, which is like a CCR, means cytochrome C reductase enzyme. Using the same principle, we have immobilized the cytochrome C reductase and detected the mechanism, how it is due to redox reaction. There will be increase in the current. For example, cytochrome bind to, there is a redox reaction, Fe2 plus 2 Fe3. As a result of redox reaction, there is a current in the electrode. If concentration of cytochrome C is more, then there will be more current response. Used two types of electrodes. One is CNT, another is gold nanoparticle self-assembled monolayer electrode. We have measured cytochrome C release in cancer cells. We have used later on, we have used screen printed electrode, because in screen printed electrode only 20 microliter is required. 20 microliter is required to fill the electrode surface. Whereas in the previous case, you need 1 ml printed electrode. We have used the same methodology for immobilization or for bioconjugation. And this is the setup for conventional cyclic voltammetric or electrochemical measurement. This is the it is portable, handheld, okay, small volume miniaturization advantages, and we have characterized with the increase in concept. The current response increases, the concentration of cytochrome C is determined, And we have also developed the cytochrome C immunosensor. Used here antigen antibody. So instead of cytochrome C in, in we have used cytochrome C antibody. We have immobilized the cytochrome C antibody on the same procedure. The similarly, so immunosensor, electrochemical super dismutase, copper zinc superoxide dismutase. By using the same procedure, we have used antibody of superoxide dismutase enzyme, and we have used by indirect method. We have detected, not by direct method, because the antibody, the one is not very much electroactive. And we have also used synthetic antibody, plastic antibody, for example, molecular imprinting polymer, element or biomimetic substance. In the presence, for example, in the presence of protein, or any small molecule analyte, the monomer is polymerized. Therefore, it will form a mold or imprint, and thereby we can selectively detect the protein. We can selectively detect the protein or any analyte. I have extensively studied in Medical College of Wisconsin on Parkinson disease, ALS disease. So recently, one project to develop a sensor for neurodegenerative biomarker. We have developed, we have studied this very to of dopamine neuron receptor. So, 16 level in neurological research. Therefore, we have developed the 16 enzyme. It use, we have shown that SOD1 enzyme oxidase enzyme. It oxidizes cysteine to hydrogen peroxide. This hydrogen peroxide is detected from the hydrogen peroxide, from the increase in current response. We have detected we have filed a patent, and we have and only 16 so this biological activity from urine. Also developed screen printed carbon electrode based biosensors for nitric oxide metabolites. We have filed patent. Commercial electrochemical which it is costing 3 lakh rupees. This is a of sample is placed here, blood or sample is placed here, and it is interfaced to. We have recently developed a virtual electrochemical analyzer to reduce the cost. In NI lab is nothing but graphical programming language. You can, before you develop, use lab only. Is so front is the 4 lakhs or lakhs. Here it is only 20,000. We have fabricated virtual electrochemical analyzer with for it. The advantage is that it can be used for any biosensors. Any biosensor can be used using this virtual electrochemical analyzer. This is the graphical user interface, graphical program languages, for example, oscilloscope, and you can develop any instrument, even EPR instrument, NMR instrument. It is actually first developed and tested, then only they will convert it into a device. So we have, based on our knowledge, we have proposed a model, handheld model, that is microcontroller based biosensor device for the measurement of nitric oxide metabolites for hypoxia disorder. We have granted money to develop biosensor. This is our proposed model. You see how we have used ARM microcontroller. This microcontroller is similar to your microprocessor, but it is very small. It is present in cars, even washing machine, TV, etc. The program is stored in. We have also stored the program. We have written a program in the code language and stored. And as you see, we have developed a portable biosensor for the detection of nitric oxide metabolites. This is the circuit diagram for the electrochemical analyzer. As I said earlier, it could be used for any sensors, for any biomarkers, any diseases. That is the advantage. Whereas if you buy a biosensor, it is only compatible for particular disease. So we have developed a virtual biosensor and USB based biosensor also. I didn't show here, and we have also collaboration with the physics lab, our physics lab. USB based. This is USB based and microcontroller based. We have used LCD monitor in so this is highly portable for hill applications. So this is the my commercial instrument. We have got the peak using commercial instrument. This you see the signals are very comparable. And we return a book entitled Biosensor so that will be useful to future generation. I have carried out extensive work. Actually, my work is on spin resonance spectroscopy, especially electron spin resonance spectroscopy. Extensively worked on spin resonance spectroscopy in college national center, and also a book on spin resonance spectroscopy. It includes BSc level to MF level to research both biosensors and bioelectronics. Both the book are and for you. I have various things and from various funding agencies, grant from various funding agencies. Recently, I'm doing out on neurodegenerative direction, and I have received some of US my US colleagues in your lab and CC, etc. I thank my first student. He is scientist research development, Cali Therapeutics, USA, California, and another student is scientist postdoc research. Madasamy was in Switzerland, now in London, and now Immunobiosensor carried out research on molecular modeling. I have worked in 3 different. Actually, I have covered only. He is now in South Korea after completing postdoc research in USA. Okay, I thank all my research scholars, current research scholars. Thank you. I have sorry for taking a lot of time. Thank you for your patience. Thank you very much. Thank you.